The last of the four major categories of biological molecules that we're going to look at are the nucleic acids, DNA and RNA. Going back to the beginning of this module, we talked about the monomers and the polymers. In this case, the monomers are nucleic acids. They're called nucleotides, and they're made up of a base, adenine, guanine, cytosine, thymine, uracil, a sugar, either ribose or deoxyribose, and a phosphate group. When we string those together and make chemical bonds in between those individual nucleotides, then we end up with a polymer, and depending on whether the sugar that is included in there in the backbone is a deoxyribose or a ribose, we call it DNA if the sugar in the backbone is a deoxyribose, and RNA if the sugar in the backbone is a ribose. Nucleotides have three parts. There's a phosphate group. This is one of the functional groups that we needed to learn. There's a ribose or deoxyribose. These are examples of pentoses because they have 5 carbons. These are carbohydrates that we learned earlier. And then bases, which are ring structures that contain nitrogen, so we called them nitrogenous bases earlier. One ring structures are called uracil, cytosine, and thymine. 2 ring structures are called adenine and guanine. The 1 ring structures are pyrimidines. The 2 ring structures are purines. Deoxyribonucleic acid, or DNA for short, is used to transmit genetic information. It's a double helix with a backbone of sugar, in this case deoxyribose, and phosphate, and then bases sticking out that can interact with one another to hold the 2 strands of the double helix together. The bases that we find in DNA are adenine, cytosine, guanine, and thymine, abbreviated naturally enough A, C, G, and T. Hydrogen bonds are what hold these bases together, and they give DNA its specificity, which we'll talk about in Module 5. Adenine forms 2 hydrogen bonds only with thymine. It doesn't bind other adenine bases. It doesn't bind C, it doesn't bind G. It only forms these 2 hydrogen bonds with thymine, because only a thymine base is the right shape to form hydrogen bonds in the right places in the right number with adenine. Similarly, cytosine forms 3 hydrogen bonds with guanine. And it's these specific base pairing arrangements that give DNA its specificity and allow it to act as the genetic code. We'll see examples of that in Module 5, as I said. A space-filling model of the DNA molecule is shown on the left-hand side. The bases here are blue. The sugar phosphate backbone is gold. And DNA has what we call major grooves, the wide space where these backbone strands are fairly far apart, and minor grooves, the narrow space where these backbone strands are fairly close together. On the right-hand side, we have a ribbon model of DNA, which helps us to see the base pairing a little bit better. Notice the presence of either 2 parallel rows of dotted lines showing the 2 hydrogen bonds between adenine and thymine, or 3 parallel rows of dotted lines showing the 3 hydrogen bonds between guanine and cytosine. The structure of DNA is shown in the attached movie. RNA is similar to DNA, but the difference is the sugar phosphate backbone has the sugar ribose instead of deoxyribose, and that's why it's called ribonucleic acid, or RNA. Another difference is that in DNA, the four bases are A, C, G, and T, T being thymine. In RNA, the four bases are A, C, G, and U, the U being uracil. Uracil and thymine are very close to one another in chemical structure, as we'll see later on, and that allows uracil and thymine to substitute for one another depending on whether we're in RNA or DNA. In other words, they have the same hydrogen bonding characteristics. Uracil binds to adenine with hydrogen bonds. Thymine binds to adenine with hydrogen bonds. In thymine, there's a methyl group along the ring structure of the base. This methyl group, CH3, is replaced with a hydrogen in uracil. But the place where the molecule makes its hydrogen bonds, shown here and here, is not affected, so both thymine and uracil have the same hydrogen bonding properties. They both bind to adenine. DNA is usually found in a double-stranded form, what we call a double helix. RNA is usually found in a single-stranded form. DNA has a deoxyribose phosphate backbone. RNA has a ribose phosphate backbone. We'll talk more about the structure and function of DNA and RNA when we get to Module 5.